So now I'm going to introduce our next speaker, which is Dr. Jean Gao. Dr. Gao is going to share with us about the genotypic characteristic of late-onset Pompe disease in Chinese. So Dr. Jean is a specialist in neurology and is currently also an associate consultant of neurology in Queen Mary Hospital, Department of Medicine. He's also the teacher of the Hong Kong University as the honorary clinical assistant professor. Dr. Gao actually obtained his medical degree, MD, from Fudan University in Shanghai in 2004 and obtained his PhD in the biochemistry and molecular genetics from Hong Kong University, Lee Ka-shing Faculty of Medicines. In 2021. Dr. Gao actually is interested and specialized in neuromuscular disease and neurogenetic disorder, and has recently actually obtained and as the first fellow in the genetics and genomic under medicine in the Hong Kong College of Physicians. He is also the council member of the Hong Kong Society of Neuromuscular Disease, and I work closely with Dr. Gao in a number of neuromuscular research projects as well. So without undue delay, it's your time, Dr. Gao. Thank you.
Thank you, Sophie. Thank you, Professor Nishino, and dear guests and colleagues. So today I will present the topics, the genetic characteristics of late-onset Pompe disease in Chinese, and summarize the major findings and some of the important studies and the most prevalent genetic mutations in late-onset Pompe disease in Chinese populations. So this is the disclaimer, and I have nothing to disclaim. The first slide shows the genetics of Pompe disease. So as you know, the Pompe disease is autosomal recessive disease, and it's a lysosomal storage disease type 2 due to the mutations in the gene called GAA, denoting the acid alpha-glucosidase, encoding the lysozyme alpha-glucosidase protein. The genetic defects leads to the deficiency of this enzyme, which can result in impaired glycogen degradation accumulation within the lysozymes and leading to the disease called Pompe. The GAA genes is actually localized to the chromosome 17, the long arm 25 to 3 regions, and it was not cloned until 1991. The gene is approximately 28 kilobases in length and encompasses 20 exons. However, interestingly, only the exon 2 start to code the amino acids, while the exon 1 and introns actually non-coding regions. So as of today, over 900 GAA variants has been reported and listed in the Pompe disease database in Netherlands, and among which around over 700 mutations actually disease causing, means likely pathogenic or pathogenic. So because Pompe disease is nowadays treatable, so we can never miss the diagnosis, and we should diagnose the patients ASAP. But what are the genetic mutation characteristics, and what are the differences between the species or between the ethnics? So it's very well known in Caucasians. The intron 1 genetic mutation called 13T to G is the most commonly seen and most prevalent mutations worldwide in the Caucasian people. It was first found and described in 1994 by Dr. Hui and has been reported by numerous literatures in the world. So this mutation actually lies in the intron 1 of the non-coding regions before the exon 2, which is the first coding exons of the first amino acids. So there are 3 different splicing forms from this MRI transcript. So first one is just slicing out some of the intron 1 with the whole exon 2 and the whole intron 2, and leading to the SV1, means splicing variance 1. The second splicing form actually is much normal. It's splicing the intron 1, intron 2, and the interrupted exon 2, and only the exon 1, exon 3 are preserved. So the third transcript actually is the completion deletion and the splicing out of the intron 1 and exon 2, partially together with the intron 2. So the 3 different transcripts may lead to different symptoms of the people, and there's a variety of the clinical manifestations, not only confined to the differences in the transcript, but affected by the age of onset, age of diagnosis, age of treatment, and duration of the diseases. So however, the genetic mutations are very different in our Asian countries compared with those found in the Caucasians. You can see in our neighbor countries in Korean, there is a report in 2013 describing 3 patients, and they reviewed another 15 patients from the previous studies, showing that the location 1316 mutation is the most commonly seen ones. This is a missense mutations change the amino acid from the MET to lysine. Another mutation is the 1857 mutations. These mutations are the most commonly seen in the Korean populations. However, because of the limitation of the patient numbers, there are only 18 numbers, these 2 mutations may not be representative, and further studies using more patient samples may be needed. The clinical manifestations of these 2 mutations are actually similar to the commonly reported late-onset Pompe diseases, including proximal weakness, respiratory failure, or very seldomly presented with cardiomyopathy. So in Japan, the landmark studies in 2007 by Dr. Fukuhara, they described 38 patients, and they found 1 synonymous mutations, 546. This mutation does change the nucleotides from the G to T, but does not change the amino acids. So this is called synonymous mutations. Initially, people think that this mutation will not be disease causing until the functional analysis showing that these mutations may have the splicing effects and change the splicing functions of the RNAs and the further protein expressions. So the clinical manifestation of these mutations have no special difference between among the other mutations. So another mutations by the Japanese group is the 1857 mutations from C to G, and this is also a missense mutations, which is similarly reported by the Korean group. However, in the Indian countries or Iran, which are also regarded as Asian, but doesn't quite look like us Asian Chinese people, Eastern Asian Chinese people, their mutations are rather different. In India, there's a report in 2021 by Dr. Thomas describing 64 people. They found that 2 mutations are mostly commonly encountered there. So the first mutation is 1933. This is a missense mutations changed to G from A and also changed the amino acids. I'm sorry, this is typo. And this mutation actually is another missense mutations, 1A to G. And the clinical manifestations actually is also the proximal weakness with some respiratory failure, but seldomly they encounter some cardiac abnormalities. However, in Iran, you see this mutation is exactly what we described in the Caucasian populations, although Iran is regarded as an Asian country. This clinical manifestation has various manifestations, from the most severe form of cardiomyopathy, respiratory failure, weakness, to the milder ones with only proximal weakness and hypercikaemia. So you see that different people, even among the Asian groups, has different manifestations due to the different genetic mutations. In Caucasians, this mutation is the most commonly seen as intronic mutation. However, in the East Asian countries, seems 1857 is more prevalent. However, in India and Iran, there are other mutations. So how about the condition in Chinese people? So this is the first description of the Chinese people genetic variants in the Taiwan in 2011. They included a 15 late-onset Pompe disease patient from 1993 to 2009. The study was performed in Taipei. The major results are the median age of onset was 15 years old, which is earlier than the Caucasian reported age, and the median age on the diagnosis was 21 years old. And 8 patients required mechanical ventilation at the age of diagnosis. The clinical manifestations include proximal muscle weakness, respiratory insufficiency, and hypercalcemia. And the mutation analysis revealed that the two dual mutations in the GAA, including 1935C2A and the 2238G2C, these are the pathogenic mutations, compound heterozygous mutations. Interestingly, you can see they also found the patient have the homozygous mutations of a pseudogene, which is called 1726G2A. So as has mentioned by Professor Geno, the pseudogenes can markedly reduce the enzyme levels in patients with Pompe diseases, especially in patients bearing the 2238 mutations. However, the pseudo-deficient was not regarded to be pathogenic ones because the patient only bearing the pseudo-deficiency genes will not have any clinical presentations. And its prevalence in normal populations is around 3.5%. That means when you find 100 people on the street, around 3 to 4 people may bear this pseudodeficiency in homozygous form, which would not be symptomatic. So in conclusion from these Taiwanese studies, that Taiwan Chinese patients with late onset Pompe disease often showed onset of symptoms in their second decade of life, with rapid disease progression, which is probably due to the specific pattern of the GAA mutation, and therefore the early diagnosis and recognition of the mutations of paramount significance. So how about the mainland Chinese? China is very large, so this study in 2004 was performed in Beijing, representing the northern Chinese population's majority. They included 27 patients, among which 9 patients are male and the rest are female. Late onset patients from 24 families, which conducted from 2003 to 2013, 10 years. And their results are more or less similar to the Taiwan populations. The median age onset is 21, in Taiwan is 15, and the median age of diagnosis is 22 years, and Taiwan is 21. And the manifestations are proximal weakness and some respiratory insufficiency and elevation of the CK. So around more than half of the patients have been found to have respiratory dysfunction, and the 73% of people may require nocturnal ventilation support at the time of diagnosis. So the genetic analysis shows the similar findings as that of Taiwan studies, which revealed 26 different mutations, including 10 that were novel. The allele frequency of the 2238G2C was to be around 30% in this patient group, which is similar compared to the Taiwan studies. Therefore, the genetic mutations in the northern mainland Chinese is also the C2238 mutation from G to C, and the clinical presentations are similar to the Taiwanese Chinese studies. The novel mutations identified in this study expand the genetic spectrum of late onset Pompe disease, and of course, some of which are not known whether it's disease causing or not at that time. And the prevalence of respiratory dysfunction may highlight the importance of monitoring the pulmonary functions in late onset Pompe disease patients, which actually we're doing always for these patients groups. So there is another study around 5 years later, also conducted in Beijing, but comprises different collaborations, comprising Beijing, Shanghai, Guangzhou, and Jinan, crossing the north, the middle, and the south of China. The patient group is increasing to 59, with 1-to-1 ratio from male to female, roughly. And the study was conducted in 2013 to 16, in 3 to 4 years. So the age of onset is similar to the Northern study results, which is around 15 years. And the median age of diagnosis is also similar, around 22 years. And the clinical presentations are also the respiratory and the skeletal muscle weakness, shortness of breath, and some nocturnal support due to respiratory failure, and the proximal muscle weakness in the extremities, and also some difficulty in walking, and also found the weakness in the trunk and upper extremity involvement as well, with the later stage lasting. So the genetic findings are also the same. They found there's GAA mutation, 2238 mutations occupies around 60%, and the rest of them are 62 missense mutation. So from this study across different provinces and cities, the most common mutations were still the 2238 mutation from G to C. So interestingly, after 2 years, there are Eastern studies that was published, which was conducted in Nanjing in Jiangsu province in 2017 to 2021. So comprising 14 patients with 1-to-1 ratio of the late onset Pompe disease. And roughly the mean age of onset and diagnosis are very similar, 15 years old and then 22 years old respectively. And the manifestations has no difference that include proximal muscle weakness, some respiratory insufficiency. Interestingly, they found that the patients from this group may present with the GI dysfunction, especially with intermittent diarrhea. So we don't know the cause. And the genetic mutation of the GAA reveals 17 different mutations, including 4 that were not novel. And still, the 2238 mutations was found to be the prevalent mutation in this patient group. So the conclusion from the Eastern Chinese studies shows that 2233 mutation is the most common mutation in Eastern mainland Chinese late onset Pompe patients. And this study was performed by Dr. Zhao in the Nanjing Medical University. So how about the condition in our Hong Kong? So this study is very famous and performed by Dr. Shing's group in 2020, yes, comprising 11 patients and 1-to-1 ratio roughly. However, 1 patient declined genetic testing, so actually genetic testing were only done for 10 patients. These patients were actually from 6 families, and the data was collected from 2000 and 2013. So the mean age of onset is relatively higher, is to around 21 years old, and the mean age of diagnosis around 28 years old. And the clinical manifestations actually similar to that was reported, including the reduced exercise tolerance and some dyspnea at night, and some weakness, especially the proximal weakness, and some fatigue. The GAA mutations revealed 7 different mutations in these 10 people, because some of the people are belonging to the same family. There's 1082 C2T and 1309 C2T, 1634 C2T, and 1935 C2T, and 2014 C2T. You can see many C2T, and also the 2238 G2C mutation. And interestingly, they found there is 4 nucleotide deletion mutations, which finally result in the frame shift mutation, the final stop codon formations, and leading to the disease in these populations. So the conclusion was the GAA 1303, 1935, and 2238 mutations are actually the most common mutations in the Hong Kong Chinese late onset Pompe patients. However, because the patients only coming from 6 families and the patient's number are rather limited, so we are not sure what exactly the most commonly seen mutations in the Hong Kong Chinese populations. And more data collected should be done to determine that. But this study also has no remarkable difference compared with the previous reported ones. So we believe the 2238 G2C mutations may be the most common mutations in the southern Chinese populations. So this figure summarizes the previous studies. In Taiwan studies, the total number is 15 people. They found this 2238 is most commonly seen. And the Northern, the Eastern, together with our Hong Kong and mixed Chinese status, also showed that this mutation is most commonly seen. So our final diagnosis and impression is the GAA 2238 G2C missense mutations is the most common mutation in the Chinese late onset patients. And the most common clinical manifestation is proximal muscle weakness, respiratory failure, and some GI upsets. So this is just a very brief introduction of the mutations, and I welcome the questions. Thank you.
So thank you very much, Dr. Gao, for the comprehensive reviews and also sharing the local data. Any question from the floor? Yes, Geno?
So a great talk. So I have a question about the most common mutation, 2238 mutation. So apparently this mutation is really widespread among the Chinese, from the north to the south, and also Taiwan as well. So can you somehow estimate how many generations ago this mutation occurred in the Chinese, your ancestors?
Yeah.
Your ancestors.
Thank you, Professor Nishino. Because you can see from these populations, there is the range of the age actually, I think it's to 47. 47 years old actually is the oldest patients among these Pompe patients. So I think at most 2 generations people will be involved in this genetic analysis.
I suspect that you have common ancestors in the whole, all patients with the same mutation. I think you have ancestors, common ancestors. So can you somehow estimate how many generations ago this mutation occurred?
Oh yeah, you mean the founder effect?
Yeah, in your ancestor, yes.
It's hard to say because the patient number are rather limited. So I would like to think around 4 to 5 at least.
It would be actually interesting to do collaboratory studies,
founder study,
other groups. Yes.
Yeah. Yes.
Can I ask one question,
yeah, sure.
Dr. Gao? For the Japanese cohort, I remember you showed there is one mutation that with no nucleotide change, no amino acid change, but is still pathogenic.
Yes.
So is there any explanation on why this happened?
Yeah. I think this reminded me of our common patient, Justin. You remember? Yeah. This mutations actually changed the nucleotide, but the amino acid was not changed. However, the changing of the nucleotide actually affected the splicing forms. The splicing site was changed, and the mRNA was abnormally spliced, leading to the stop codon and the truncated mRNA transcription and the final protein translations. So that may be the mechanism for diseases. So traditionally, we only think that the missense mutation or some other, we always think that synonymous mutations will not be disease causing most of the time. And by the whole exome sequencings, we may miss out some of the synonymous mutation because we think that does not change the proteins. But nowadays, we think that condition is more and more commonly seen, especially for those undiagnosed neuromuscular diseases. So my explanation is that although it does not change the protein, however, the changing of the nucleotides will affect the splicing, leading to the disease.
So this is an intronic mutation?
This is not an intronic mutation.
No.
This is an exon mutation. However, the mutation changed the splicing site, leading to the aberrant splicing of the mRNA.
So there's different isoforms that cause
yes.
Oh, anything? Professor Nishino, can you supplement?
Yeah, so I agree. So I was not involved in this study, but to prove that the aberrant splicing you need, you may want to do RNA sequencing using the affected sample, in this case muscle biopsy sample. So for this, actually muscle biopsy is also useful. And we recently actually published a paper together with my collaborators. So they actually developed new prediction software, which is called Splice AI. This is very useful, actually. So with this type, especially for this type of synonymous or variant, you can just put the information, then you can, it actually shows really, prediction with really high accuracy. And I didn't check this particular variants, but most likely it will show the new, the creation of new acceptor site or donor site, which, causes the aberrant splicing.
I see. Yeah. Thank you very much.
Thank you.
Thank you very much, Dr. Go and Professor Nishino.